Hello students, once again welcome back to our biology class. Dr. Raghavendra here. In the last class we were discussing about DNA fingerprinting. Yes, we studied about the DNA fingerprinting as such. It is developed by Alex Jeffrey in 1984 when he was studying about the sequences of myoglobin. And then mainly the DNA fingerprinting is studied based on the 0.01% variation. What is that 0.01%? There are 3 into 10 raised to 9, that much huge number of DNA base pairs, and 99.9% DNA is very much the same in all the human. Then how to identify the uniqueness of the or differences between a person to person? Yes, it is done mainly by targeting the satellite sequence, we call it as or repetitive sequence we call, or we also call them as VNTR, variable number of tandem repeats. So for that we need to understand the isolation of the DNA. Yes, we studied the significance of the DNA, that the fingerprinting. Samples such as maybe the hair sample, maybe the bone or blood sample, and maybe the saliva with the buccal epithelial cells, or it may be what you can say the semen sample or skin cells. Anything can be taken and DNA isolated. So that is what we say mainly the significance. DNA can be classified as here mainly the bulk DNA as well as the sat DNA or satellite DNA. Satellite DNA forms nearly 10% of the bulk DNA after cutting the DNA with the restriction endonuclease enzymes. And then, so this satellite DNA has a repetitive sequence, we call them as a mini satellites and microsatellites. So microsatellites are hardly 2 to 6 base pairs are repeated, maybe several thousand times also. And mini satellites 10 to 60 bases are repetitive. Again, they may be of millions of times repetition. So this kind of repetition pattern is unique, differ from person to person. So that only the DNA fingerprint is targeting. Well, so we have discussed mainly why these kind of variations. Yes, when the variations exist, it may be more than 0.01%. So the reason behind is mutation. Mutations brings lots of genetic variations. It is the reason for evolution, reason for speciation also. Mutations keep on continuing generation after generation. Yes, it is mainly what you can say expressed well. So we have discussed all about this, and again mainly the DNA fingerprint requires the better sophisticated method of isolation and proper conduction of the steps. Let us understand the procedure of DNA fingerprinting. There are hardly 5 to 6 important steps involved in the DNA fingerprinting procedure. So steps in the DNA fingerprinting. Well, what are those steps? Number 1, isolation of DNA. Number 2, it is mainly digestion of DNA. So how to digest the DNA? Yes, to cut the DNA by using the restriction endonuclease. We say digestion of DNA by REN, restriction endonuclease enzymes specific. Then electrophoresis is a method to separate the DNA fragments based on their charge and molecular weight. Number 4, alkali treatment to the separated or isolated DNA so it can become single stranded. Number 5, there is what you can say blotting DNA. It is also called as southern blotting technique. Okay, blotting the DNA on nitrocellulose paper. Number 6, Hybridization. Hybridization, making the DNA hybrid DNA. Number 7. Okay, yes, autoradiography. Radiography, yes, to carry over or to make the DNA fingerprint on the X-ray sheet. So these are the few important yes steps involved in the DNA fingerprinting method. I hope you got an idea. Let me tell you now the DNA sample. Yes, from where we can isolate the DNA sample. Yes, it may be any kind of a body cell. So as we have seen, it may be mainly can be isolated from the saliva having the buccal epithelial cells. Buccal epithelial cells have the nucleus, or maybe skin cell, or maybe WBC, or maybe the bone cells, or maybe the sperm sample also. Yes, so number of cells can be utilized mainly to isolate the DNA. So yes, isolation of DNA mainly involves gradient centrifugation. Yes, what we do, tissue, okay, or the cells, digestion, okay, and then what we say that the first step, tissue or the cells digestion is done by using particular buffer, by using particular kind of chemical, by using the TCA, also we can say trichloroacetic acid, by using TCA or trichloroacetic acid also can be used. So further what will happen, yes, we will get the DNA. So we will get the DNA which is a huge bulk mass. Okay, so we call it as mainly the bulk DNA. Bulk DNA we get. So this is further what we have to do, gradient centrifugation when you carry. Yes, you will get, and you have to add enzyme restriction endonuclease. Okay, so finally what you will get, you will get the DNA sample digested well. So the DNA sample now got fragmented. Yes, DNA sample got fragmented. So this is, we get the fragmented DNA pieces. Okay, so thus we get the DNA. Once we treat the bulk DNA with the restriction endonuclease, apply the gradient centrifugation, and you will find mainly the yes DNA fragments of what we can say mainly. So this DNA fragments, addition of restriction endonuclease itself is the second step. Digestion of the DNA, we say digestion of DNA, what, by using the restriction endonuclease, and you get the fragmented pieces of the DNA. Now you need to go further for the next step. What is that? You need to go for the third step. What it is? Electrophoresis. So the third step is electrophoresis. What is this? Separation of the biomolecules based on their molecular weight, okay, under the influence of electric field in a porous gel. Okay, well, so here what we are observing mainly, so the DNA sample is here we added. So this is what we say the negative electrode, here is the positive electrode, and these are the DNA, and yes, it is the gel sheet which is porous by nature, and it is now kept in the buffer tank. So buffer is filled, okay, then current is mainly given. So finally what happens, there is a movement of what we can say that movement of the DNA. Okay, yes. So we can say that the DNA at this high molecular weight DNA, which remains there only, it can't migrate, it can't move. But whereas the more low molecular weight DNA migrate, low molecular weight DNA will move faster and lower side. So DNA being a negatively charged, it has high affinity towards the positive electrode, so it keep moving into the porous gel under the electric field. The separation of the DNA based on their molecular weight and size, we call it as a electrophoresis. So now electrophoresis is done. So once we get the gel sheet outside, yes, the fourth step here, we have to take out that gel sheet which has nearly around 3 DNA samples we have added over there. So here the gel sheet is taken out. So this is the gel sheet and it has what we can say that DNA fragments. So here that has the DNA fragments. So here what we find, these are all the double stranded DNA. Okay, 1, 2, 3, 4 and 5 bands. 1, 2, 3, 4 and 5 bands. So these are mainly the gel sheet where it is kept in a container. So it is kept in a container and it is filled with alkali solution. We call it as a alkali treatment. Okay, so the double strand DNA will break. So then the double strand DNA, their hydrogen bonds will break and then it become, yes, single strand DNA. It will become after alkali treatment, alkaline solution will break the double bond, mainly the hydrogen bond between the nitrogen bases of the DNA, then DNA become single strand. This is a very, very important and crucial, what we can say, the step. So after treatment, this what we will do. So here the fifth step, what is that? Mainly the southern blotting, we say. Southern blotting. What is the use of this southern blotting? So here is the gel sheet, right? So this is the gel sheet. Now above this they are going to keep the nitrocellulose paper and then 2, 3, 4 and 5. 1, 2, 3, 4 and 5. 1, 2, 3, 4 and 5. Yes. So the gel and then nitrocellulose paper. So the nitrocellulose paper keep on absorbing everything what is there on the gel sheet. So single strand DNA now transferred on the nitrocellulose paper. DNA, single strand DNA, yes, DNA transferred on, yes, nitrocellulose paper. So nitrocellulose paper get that DNA, whatever the DNA now coming on the nitrocellulose paper, it is a single strand DNA. So this single strand DNA now we have to convert it into a hybrid DNA. How it is possible? Yes, it is possible. We call that the technique as hybridization. The sixth step is hybridization. Hybridization means what? So single strand DNA, yes, 1, 2, 3, 4 and 5. 1, 2, 3, 4 and 5. 1, 2, 3, 4 and 5. These are all single strand DNA, but what they will do? Yes, they spray mainly what we can say that yes, radio labeled nucleotides added. So then it become radioactive DNA or hybrid DNA. Okay. So what will happen? It become a double stranded DNA, or now they become yes the hybrid radio labeled DNA. That means imagine this is the DNA. So here we have adenine, we have thymine, we have guanine, we have cytosine. What should match here? To this thymine, to this adenine, to this cytosine will match, to this guanine will match, but they are labeled with the P32 like this. P32. So P32, P32. So radioactive tagged the bases are added. So one strand is normal, other strand is a radioactive labeled strand. That's why whenever a light ray pass through this, they reflect back. So this nature is very, very important. Hybridization is very important to make further steps. So what is the last step? We call autoradiography. Yes. So let me mention what actually happens during the autoradiography. I hope you are getting the idea why they are done the radio labeling. Yes, to ensure that whatever the DNA, yes, there on the nitrocellulose paper, that should be able to reflect the light. So this property is very essential at the time of autoradiography. Let me mention here autoradiography method, what they do. So during autoradiography, yes, they will keep a X-ray sheet. Okay, mainly they will keep an X-ray sheet on the what we can say mainly this nitrocellulose paper with a hybrid DNA. So seventh step, let us understand autoradiography. Yes. In this what they do, yes, here imagine we have our nitrocellulose paper with the hybrid DNA. Okay, yes, we have our hybrid DNA here. Fine. Generally at a higher level there is a high molecular weight DNA. Yes. So about this they will keep mainly, yes, this is the nitrocellulose paper, nitrocellulose sheet with hybrid DNA. Why? Because imagine here this is the DNA, it has adenine but labeled with the P32. Now it is a radio labeled. Here it has a guanine but labeled with the P32 like this. Adenine will pair with the thymine, guanine will pair with the cytosine. So this strand is radio labeled and it reflects the light out. So there is a nitrocellulose paper which carry the hybrid DNA, and above that they have kept the X-ray sheet. Now they will pass the X-rays. Okay. Yes. So X-ray exposure. But all the X-ray may pass through this, but at this point wherever the radioactivity is there, they reflect back. Otherwise, so finally we get DNA fingerprint. So we get mainly on the X-ray sheet the patches 1, 2, 3, 4 and 5. 1, 2, 3, 4, 5. 1, 2, 3, 4 and 5. These are DNA fingerprint on the X-ray sheet. So these are the DNA bands or fingerprint. DNA fingerprint on X-ray sheet. This is what we say the whole technique of DNA fingerprinting. I hope you are understanding this. Let me explain it once again. So the first step of DNA fingerprinting is mainly isolation of DNA. So DNA is not always available at the top of the cell or outside the cell, it is inside the cell. We need to we need to get a better technique to isolate this DNA. So DNA isolation mainly either from the tissues or the cells collected. Usually if the tissue is there, to disintegrate that tissue, yes, to separate the cells we are using trichloroacetic acid. So otherwise, yes, when the cells are already separated, there is no much of the usage of this kind of acidic compound. Certain buffers are used to take out the DNA. So when we mix the particular kind of buffers and then we treat the cells with the particular amount of ethanol and the chloroform, and then yes, we are able to break those cells, we are able to break the nuclear membrane, we are able to take out the DNA. So when the DNA is collected, it appears to be the cottony mass, we call it the bulk DNA. So we need to add the restriction endonuclease to this, because all the unwanted bulk DNA we don't want. We are interested in a particular repetitive sequences only. So we will add the restriction endonucleases, that is mainly to digest the DNA, and then yes, gradient centrifugation we are applying. Then so the spinning speed we call it as for the first 5 minutes maybe 10,000 rpm, second 20 minutes maybe 50,000 rpm, next it may be 75,000 or 1 lakh rpm. Like that the centrifugation speed will change, we call it the gradient centrifugation, and then we find the fragments of the DNA. So these fragments of the DNA are collected and then put into the gel electrophoresis. So based on their molecular weight and charge, the DNA will separate as a different bands. Then this gel sheet is taken out. Yes, the gel sheet is also stained with a particular stain, and then this gel sheet having double strand DNA treated with alkali solution, the double strand DNA will become single strand. It is essential step because we are going to convert this double strand into hybrid DNA. So now what we say that the DNA now can be taken up on the nitrocellulose paper, which is the absorbent paper. So the taking of the DNA outside from the gel to the nitrocellulose paper transferring is called as a southern blotting. So after doing the blotting technique, to this paper which is already having single strand DNA, we will spray with the radio labeled nucleotides. Now after spraying, they will again as a complementary, yes, they will attach, and then again it become double strand DNA here. So this double strand DNA is a hybrid DNA because it is a radioactive now. So because radio labeled nitrogen bases we have added, the radio labeled adenine will pair with the thymine, guanine will pair with the cytosine, radio labeled cytosine will pair with the guanine, radio labeled thymine will pair with the adenine. So like this, hybrid DNA is formed on the nitrocellulose paper, and above that paper we are keeping the X-ray sheet. We are exposing this total set to the X-rays. X-ray will definitely pass through all the part except wherever double strand DNA there, reflection happens. When the reflection happens, now the white patches or banding pattern will appear on the X-ray sheet. This is called as a DNA fingerprint or a DNA banding pattern. So DNA banding pattern is very much unique. It differs from person to person. So thus the DNA banding pattern can be comparable, can be analyzable, can be accessible, so that easily we can compare with the person to person, and then we can identify number of things. So that all comes under applications of the DNA fingerprint. Let us understand that also now. So when we are talking about applications of the DNA fingerprinting, I hope you got this idea regarding the different steps involved in the DNA fingerprinting. Now let us go ahead here. What are the major applications of DNA fingerprint? So DNA fingerprinting is helpful mainly to resolve a number of medico-legal complications or parentage cases. So applications of the DNA fingerprinting. Number 1, to resolve number of medico-legal medico-legal cases, that includes paternity and maternity cases. Maternity detection, we can say. Now, second, anthropogenic studies. Studies mainly evolution and human races. Okay. Then number 3, pedigree confirmation. Pedigree confirmation, population migration, okay, population genetics. Yes. Number 4, confirming gene expression in GMOs, genetically modified organisms. Yes. Number 5. So mainly what we say that yes, to resolve number of medical legal cases, anthropogenic studies, and then what we have studied mainly here, what regarding the pedigree analysis also, and then we are able to understand mainly confirming the gene expressions in the GMOs and forensic studies. Forensic studies to solve murder and rape cases. Okay, so these are the different kind of applications what actually the DNA fingerprint help us to resolve the number of medical legal cases such as paternity and maternity cases. Imagine, yes, here we can say that the sample of, so let me mention, this is a DNA profile. So this is the child and the mother, okay, person 1 and person 2. So now child has a DNA fingerprint like this, okay, so 1, 2, 3 and 4 banding pattern. So yes, mother's banding pattern matches with the child, definitely. So now the person 1, yes, has a different banding pattern. Then person 2 also has a different banding pattern, we can say that. So here you can find this one band, another band matches with the child's DNA profile. So the person 2 is the father for the child. So a woman is raped by 2 people and then she don't know who is the actual father. Then at that time, yes, medical legal or parentage cases or maternity and paternity cases are resolved very easily by using the DNA profiling or DNA fingerprinting. And then anthropogenic studies means a human evolved, yes, mainly human earlier time, yes, in the evolution course they migrated to the Africa, Asia as well as to the Europe nations. So what is the percentage of DNA similarity between the people of different continents? Yes, they can be studied well. And then keeping the DNA fingerprint profiling, yes, at the time of immigration also, people move from one country to other country, they go for the facial surgery and in somebody's name they may go, but when we catch them, yes, we can catch them by using the DNA profiling also. And then pedigree confirmation, yes, the number of characters are expressed generation after generation, and the further the confirmation, yes, this is the so and so type of disorder that occurred in grandparents, parents and next generation also. By using the DNA profiling, we can confirm it. Even population genetics, population migration, population variation also can be studied very well by using DNA fingerprinting. Confirming the gene expression in genetically modified organisms. Yes, when you do the DNA profiling of the human, DNA profiling of an animal, it may be the cow or the sheep or the goat, that says that human milk proteins are there, human therapeutic proteins are there, means that human DNA should be there in that organism. So by identifying the DNA similar bands in the human and plant or animal, you can confirm, yes, this is the animal totally undergone GMO or genetic modification. Forensic studies to resolve the murder and rape cases also, DNA fingerprinting helps a lot. These are the most important and major applications of the DNA fingerprinting. Yes, thus we are done with the overall discussion of the DNA fingerprinting also. In the next class with some more new concepts, new chapters, we will meet again. Till then, take care, study well. Thank you very much.